Okay, good afternoon, everybody. Thank you for attending today's seminar presentation on a crisp Manitoba day. I'm glad you made it out here. It's really my pleasure to introduce you to today's speaker, Dr. Chengbo Yang. Chengbo is an associate professor in livestock nutrition and nutritional biochemistry in the Department of Animal Science here at the U of M. Chengbo obtained his PhD in monogastric animal nutrition at the University of Guelph in 2011. He joined the University of Manitoba in 2016 after about 5 years working in industry. He's currently conducting research in the area of gut health and nutrient utilization relevant to non-ruminants. He has acquired about $10 million in research funding as principal investigator or co-principal investigator from NSERC, CFI, Canada Foundation for Innovation, USDA, the Swine Cluster, and other agencies. He currently serves as an editorial board member for 2 journals, including the Canadian Journal of Animal Science. He has provided training to over 50 highly qualified personnel and has published over 90 peer-reviewed articles. Chengbo received the 2018 Canadian Society of Animal Science Young Scientist Award and the 2018 Terry Falconer Memorial RH Institute Foundation Emerging Researcher Award. He also received a 2019 Merit Award in Research, Scholarly Work, and Creative Activities at the University of Manitoba. So without further ado, the floor is yours, Chengbo.
Yeah, thank you very much for the introduction, Nazim. And good afternoon, everyone. And thanks for coming for my presentation, especially it's very cold today. And also thanks the opportunity to share our research result with you. So today my presentation title is more than just chicken feed and the antibiotic alternative in poultry diet. So actually, as I mentioned mentioned it to some people, I actually did the presentation on November 7 in the US at St. Louis just exactly 1 month ago. And so before that, I want to give you some figure about the industry. So when you look at this figure, this is the global animal feed market forecast. So this the 2021, the total feed market value at the $482.1 billion. So this is a lot of money. It's a big industry. So by the 2027, the total global feed market value will be about $589.4 billion. So you can see the compound annual growth rate is about 3.5%. Actually, it's very good. So if you think about the average, the global economic growth rate, the 2021, the 5% more or less, and the 2020 just negative 3.8, something like that. So the 3.5 actually quite good. So it's a very dynamic industry. And then actually another figure, so about a forecast of the global feed additive market value. So the 2021, there's $37.4 billion for the globally, and in the 2027 will be about $56 billion. So and you look at the increase rate, compound, the annual growth rate is 8.1. It's a almost double 3.5, right? More than double, actually. And also you look at the share of the feed additive in the total feed value. It's from last year 7.7% the share, and by the 2027 it's 9.5%. That means that the increase rate of or growth rate of the feed additive value is faster than the animal feed. So there may be some reason for that. So the potential, the reason for the faster growth of the feed additive in the globally. So there are several key drivers. The first one is increased milk product consumption, especially Asia Pacific area, the 40% increase. So this is the major driver to increase animal feed and also increase animal feed additive. And another reason is antibiotic ban or restriction. So, and they're looking for more antibiotic alternatives, for example, in the European Union, China, Canada, and the US. So this is the increased demand for the feed additive as well. Also, another reason, the favorable regulatory norm, for example, carbon neutral and the carbon footprint. So that means the industry needed to look for strategy to increase the efficiency, right? Reduce the waste, reduce the carbon neutral footprint. So that's also is a driver for the increased feed additive. And also the increased livestock disease outbreaks, for example, in the poultry industry, the white striping and the woody meat. So this is a big concern in the US poultry industry. In Canada, not the big concern because we are not promoting heavy births. So these are potential reasons to drive the growth of feed additive. So one of the important contributor is antibiotic alternatives. So today is a major topic for my presentation today. And back to the poultry industry. So this is the figure, look at the annual chicken meat production. As you know, chicken meat is a major meat for humans, right? And they followed by pork and the beef, right? So you look at the actually production and the forecast from the 1950 to 2050, right? So you look at by 2008, 18 million tons of poultry meat, and by 2040, 160 million tons, and the 2050, 200 million tons. So the increase is very dramatic. And because the demand is supplied by the intensive poultry farming system, so there you can see this basically is the setup in the poultry barn looks like, and so they have this intensive poultry farming system has a lot of challenge. I think one is because they can bring a lot of stress. For example, the stress can reduce feed efficiency, reduce the body weight, and also increase mortality. Okay, this is one stress, one challenge. Another one is a pathogenic infection. So the two major of pathogens, for example, the coccidiosis is a big concern for poultry industry, and also another necrotic enteritis. So the major pathogen is Clostridium perfringens and also E. coli as well. So this is a big challenge for the poultry industry. So I give you a little more detail about the Clostridium perfringens, and this is the necrotic enteritis is the one of the major pathogen is Clostridium perfringens. This produce the letter P toxin, and the symptoms are like a decreased appetite and the severe depression, dark color diarrhea, closed eyes, and ruffled feathers. So this disease leading a huge economic loss. For example, about over $5 to $6 billion annually worldwide. It's a huge loss for the industry. And also give you a little bit background about the E. coli in the poultry. There are two types of E. coli in the poultry. One is non-pathogenic E. coli, and the other one is pathogenic E. coli. So non-pathogenic E. coli, they can also provide the nutrients, synthesize some vitamin, right? And today I will focus more like a pathogenic E. coli. So there are two types of the pathogenic E. coli. One is located in intestine, called intestinal pathogenic E. coli, APEC, and it causes diarrhea. And another one is extra-intestinal pathogenic E. coli, like extraPET. So it can cause cholebacteriosis. So this E. coli from poultry can cause the disease in poultry, but also can harbor the antimicrobial resistant gene. And the exapac from poultry may have a potential to infect the humans, okay? So just to give it a little bit background about these two pathogens. And so the challenge, I think, the antibiotics, in-feed antibiotics, has been used to copy this, to prevent this infection. So the in-feed antibiotics, so they have different function, interrupt the bacteria protein synthesis, and they also memory. And I don't give more detail about the mechanism for the functional antibiotics. So the purpose of the antibiotic used in the feed is the one is a therapeutic treatment. So once the disease outbreak, so they use the antibiotic for the treatment, and they also for preventive treatment put in the feed. So also they are for the growth promoter. So usually, conventional feeding system have better growth performance than the no antibiotic system. However, because of the use of feed antibiotic in feed, that can cause potential antimicrobial resistance by different mechanisms, for example, antimicrobial degradation, and modify the drug target, and also reduce the intake of antibiotics, or increase the bump of the antibiotics, okay? So they can also cause the prevalence of antimicrobial resistance. And they also can spread the antimicrobial resistance from different way for vertical transmission or horizontal transmission. So this can potentially pose this threat for the industry and also for the human health. So because of this, so there are a lot of pressure from the consumer and from the public. So the pushback for the industry. So you can see that a lot of companies that expected they want a food system, assume food is safe, affordable, and healthful and sustainable. You can see the major players like Tyson Food, Cargill, Costco, and the Chicken Fillet, and McDonald's, and the Pizza Hut, and the Subway. They are promoting or advocating the responsible use of antibiotics. So this actually to push back the industry have to remove or reduce the use of antibiotics. So it's a huge pressure from the public. Yeah, for this reason, and the industry also take the government take a lot of steps to do that. So you look at here, and in the European Union, they have banned the in-feed antibiotics on the 2006. So it's much advanced. And in Canada, so category 1 drug really is very important, very high important for human. So for example, like Cetaphil, this antibiotics is very high important for human. That was cannot be used in 2014, and category 2 drugs by 2018, for example, like penicillin. This is high important for human. And the category 3 that cannot be used by 2020 is, for example, bezetration that was used in the poultry industry a lot. So now we cannot use. So this also in China and Nigeria, and Africa. So these are the regulatory pressure. And because of that, so we remove the antibiotic from in the feed or from the feeding system. So we have some challenge, because challenge is still there. We need this alternative of antibiotic to help the industry to copy this challenge, right? So, and the antibiotic alternative is not a new topic. There are so many types of alternatives. For example, probiotics and the prebiotics and the vaccine, is a major way to prevent the disease as well. And the essential oil. So today we use the essential oil as an example to demonstrate how this helps the industry, can help industry to have an antibiotic-free production. So because the new antibiotic development is very slow, and there are more antibiotic resistant it developed very fast, right? So in this case, alternatives. So today we talk about the essential oils. So essential oils, the definition of essential oils is comes from the plant. It's concentrated hydrophobic liquid containing and aroma compounds. It's from the plants. But also essential oil can be synthetic, natural, identical, right? Can be manufactured. So the essential oil has different properties, and including anti-inflammatory property, the oxidative property, and also can inhibit the colon sensing, so can interrupt the communication within the bacteria, okay? And also has antimicrobial property. So this a lot of function for the essential oil. So today we are going to just focus on two types of essential oil. One is cinnamaldehyde. This cinnamaldehyde, this is the molecule. They have aromatic ring. So it's they're here. And another one is Cicha. Cicha is another type of essential oil. They have two formats. One is Cicha A and Cicha B. So they're slightly different in the molecular structure. And these two actually are approved used in feed in Canada. So, but they use the industries already have a lot of successful product about essential oil. But it also has a disadvantage of the essential oil application. And because the one is not stable, because volatile compound, right? Not stable during the storage and the heat processing, for example, feed pelleting process, the high temperature, right, can reduce the stability and the gastric transition. So this usually can be absorbed quickly in the stomach. So you want the essential oil to reach the lower gut, for example, small intestine or large intestine, right? And because the aroma compounds, so low concentration maybe can increase palatability, but the too strong the smell also can reduce the palatability. So there's a major disadvantage. So there are strategies to overcome this advantage. One is like a macroencapsulation. So this can increase the shelf life of essential oil during the storage and also improve the stability during the heat processing, gastric transition, and remain the palatability. So for this benefit. So the industries also have they already have some successful products use macroencapsulation. And they are used different career protection method. For example, they can use protein, can use the fat, or use this polysaccharide to protect essential oil. For today's presentation, for our research, we use these two ingredients, like soy protein, polysaccharide, and the soy protein, two compounds, okay? So this macroencapsulation was developed in the Gulf. So I'd briefly to talk about the procedure, how to encapsulate this essential oil. Basically, they use a Maillard reaction to put the soy protein and this soluble polysaccharide together and make the Maillard reaction to product. And because this can protect the essential oil better than just a mix. So you need to go through the soy protein polysaccharide Maillard reaction. So this protein can be soy protein, but can also come from animal protein, for example, milk protein, whey protein as well. So because for organic production, so cannot use animal protein, right? So in this purpose, you can use the soy protein. If it's not organic production, so you can use the protein from the animal, for example, milk protein, whey powder, right? So after they make the Maillard reaction compounds, and then you just do the immersion to put together, and then make the liquid the homogenization. So in this case, you can use this as a liquid format to add in the water, or you can use the feed. You can further to use the spray dry to make it a powder, and later on you can add in the feed. That depends on the application. So actually, the well-known Canadian companies already registered in the Health Canada already registered 2 products. One is with animal protein, another with plant protein as a carrier. Okay. So this I don't want to spend more time on the microencapsulation. So, and then there are just research gap because the previous study already demonstrated that suture encapsulated with SPPMP can be used to control necrotic enteritis in the broiler chicken in 2016. So another study also encouraged the potential use of encapsulated cinnamaldehyde for control animal enteric passaging for oral in-feed administration. So, however, the further study are still needed to evaluate the efficacy of the encapsulated tritia and cinnamaldehyde as antibiotic alternative, and then to elucidate the underlying mechanism in broiler chickens. So in this project, we hypothesize that the encapsulated suture around or combination with cinnamaldehyde can replace the in-feed antibiotics through the controlling the electrical NE, improving the intestinal digestive absorptive function, and the modulating gut microbiota and improving meat quality in broiler chickens. And also encapsulated suture and the cinnamaldehyde can also reduce the prevalence of antimicrobial resistance and the virus of the chicken fecal E. coli, which may reduce the risk impact for infection in human. Okay, so not only just help the industry, but also reduce the potential risk for the human as well. So based on the hypothesis, we set up the objective. First of all, we investigate the effect of the protected essential oil on the chicken growth performance and the gut lesions and the fecal microbiota. And this is study 1. And study 2, we also look at the antimicrobial resistant phenotype and the genotype and the various E. coli isolated from the chicken fecal sample. And also we look at the intestinal digestive, absorptive function and meat quality and the gut microbiota in the broiler chicken. This is study 3. So this is study 4. So we establish C. elegans, this is a worm, C. elegans lifespan assay on the actual pack to know where the model is successful predict pathogenicity of the chicken E. coli, pack E. coli. So in the study 1, so we look at this published in the Poultry Science. So basically this study was conducted in Quebec. So we have two groups, one group with vaccinated with coccidias, another group 2 non-vaccination. So, and then for each group, we have 5 treatments. So we have a control diet, no antibiotics. So this the positive control use the BSE. By that time, so we still can use the pacitrizin. So that's we use that's antibiotic as positive control. And then with cinnamaldehyde 100 ppm, suture 100 ppm, and the combination at 100 ppm. With the, and this we look at the starter and grower finisher about 28 days. Okay, and we also check the gut lesions and the cecal digester collection, and they also growth performance for these 3 phases. So for gut lesions, we distinguish the lesion come from the NE and also from coccidias. So the NE score we can, according to this 0 to 3 score system, and the coccidias score use the 0 to 4, and also based on different Emeria species, okay, because they're different. So for this scoring, so we cannot do ourselves. We ask the vet specialist because it's very confusing. So if you don't have experience, it cannot make accurate scoring. So we have the vet specialist to do this, and then we for extracting DNA from the microbiota, and they do 16S RNA sequence. So when look at the growth performance, so basically you want to vaccinate birds or the non-vaccinated birds. So vaccinated birds had non-vaccinated birds had better growth performance than the vaccinated birds. Okay, because the vaccination maybe also cost energy, can reduce the growth performance. So and then look at the antibiotic treatment and the essential oil. So from here you can see basically the antibiotic group is best, okay, compared to control, and the essential oil supplementation can bring the similar growth performance compared to the antibiotic treatment, okay. And then we look at the gut relations. So from this figure, you can see the vaccinated birds had a higher coccidius relation score due to the E. acetivaria and the E. maxima. So because the vaccination, that is normal to see that. And they also, from this figure, you can see the higher NE relations while showing the vaccinated birds, but the lower in the birds fed cinnamaldehyde and the cinnamaldehyde citrate compilation. So which means the essential oil can reduce the relation in the gut. And they also look at the second microbiota. So lactobacillus at the Clostridium were held abundant in the non-vaccinated birds. Lactobacillus was more abundant in the bird fed with citra, while the Clostridia and the Enterococcus was less abundant in the bird fed essential oil and the essential oil compilation. So which means the essential oil can increase the lactobacillus. So they consider as beneficial bacteria for the host, for the birds, right? And the Clostridia and the Enterococcus considered not beneficial for the host. So this essential oil can change the microbiota in the sequence. So based on this study one, we can summarize this encapsulated essential oil can improve the general growth performance and similar with the bacitracin, this antibiotic treatment. And the encapsulated essential oil and the compilation has a similar effect on controlling NE due to the Clostridium perfringens and coccidiosis caused by Emilia species. And the combination of the vaccine encapsulated the essential oil can modulate the composition second microbiota by increase the beneficial bacteria lactobacillus and decrease the Enterococcus and the Clostridium. So this from this study, the summary. So, and we consider the second study, so use the same animal study. So this, we want to look at antimicrobial resistant genotype or phenotype. So with the supplementation of the essential oil. So the animal study the same. So this paper was published in Journal Food Protection. So, and you can see there, basically first step, we isolate E. coli from chicken feces, okay, from different animal group. So there's animal study from the first study. And then we store until we analyze the sample. So we did two parts. One is do the antimicrobial susceptibility check and also whole genome sequence to compare the different strain of the E. coli. So basically, this uses the sensitized automatic system to measure the antimicrobial resistance and against the measure MIC, the minimal inhibitory concentration of 14 antimicrobial in the old isolate, according to the clinical laboratory standards. And genome DNA was extracted from the 240 isolate. That's a lot of work there. And they use the Illumina me sequence platform to sequence. So we'll look at the phenotype first. So when you look at here, the citra and the cinnamaldehyde and the cinnamaldehyde citra combination could reduce the resistance level to the amoxicillin, safety 4, and the ditamycin, stepmycin. So reduce the resistance level, okay? And also resistance level to the alomycin, as these antibiotics, could be decreased with the increased age. It's very interesting. With the increased age of birth, the resistance level is reduced. This potential may be once with the increased age, the microbiota in the gut, in the birth gut, is more stable. But the exact mechanism we don't know. So and then we look at the genotype. So do the sequence. So prevalence antimicrobial resistant gene. So you can I don't want to go through too much the table. So you can see different type of the resistant gene. So cinnamaldehyde, the switcher combination reduce the prevalence antimicrobial resistant gene to this 1, 2, 3, 4 group of the antimicrobial, like beta-lactam drug and the tetracycline. So reduce this resistant gene, okay? And the prevalence the resistant gene could be affected by age of birth as well. So sometimes, most times it can increase, also it's not just increase, but sometimes increase. So this age also can affect the prevalence of the antimicrobial resistant genes. So, and then we talked the plasmid, because plasmid also plays an important role for the antimicrobial resistant transmission, right? Horizontal, vertical transmission. So we do this one sequence to look at this data. And so results show the essential oil reduce the prevalence, the most detect the resistant plasmid. And but however, this is not affected by the essential oil supplementation. But the prevalence of the resistant plasmid could be affected by the age of birth as well. So this is the same. It looks like the age of the birth as the big factor to affect the antimicrobial resistance. And so this virus gene, so the essential oil treatment did not affect the number of the virus gene expression, and the number of the virus gene and the lower in the day 6 compared to the day 16, 23 day, and 27 day. So which means with the increased age of the birth, so the virus gene actually expression is higher. So also the meticulous sequence typing, this is also very important to screening the antimicrobial resistance. So we look at here, the concentration and the cinnamaldehyde and the citral and the combination reduce number of the ST38 significant compared to the NC. So these ST38 are related to the avian clubs of the human infection. And the higher number of the ST38, 117, and day 23 day compared to the day 6, 16, 27. This is similar with the virus gene as well. So this is increase the age, they have a higher the ST38. So we also make the phylogenetic tree and compare the different isolation. So we found that a lot of isolate that is similar to the human except. So for example, there are 25 isolates are highly related to the human except. So from different groups, the control and the antibiotic group and the essential supplementation group. And there's 6 serotypes, so they're also related to the human aspect. And the 4 MLST sets are related to human aspect. So this means this isolate from the poultry can potentially have to infect the humans, can bring the public health risk, right? So this, in order to do that, we later on we are doing a further study to verify that. So based on this study, we can summarize that the dietary selamaldehyde affected the MR phenotype and also genotype of E. coli in the chicken feces. So not only to control the disease, but also can affect the phenotype or genotype of antimicrobial resistance. So this basically can reduce the risk of the antimicrobial resistant development. And the birth age could also be a factor that affect MR phenotype or genotypes of chicken E. coli, fecal E. coli, appear to influence the number of the various gene serotypes and also MSTs. So this is very interesting, but we don't know what the exact mechanism yet, but it looks like the age is the big factor to affect the AMR phenotype or genotype. And also dietary essential selamaldehyde affected the presence of the different plasmid harboring antimicrobial resistant gene in the studied E. coli, so indicating the ability of this essential to decrease potential antibiotic resistant gene transfer between the bacteria and the other chicken gut bacteria. So this essential can reduce the risk. So the study 3, so we want to look at how this affected the growth performance and the digestive and absorptive function, and also meat quality, because we mentioned earlier that decrease the disease outbreak like white striping, woody meat, right? So we look at the meat quality as well, and the gut microbiota in broiled chicken. So the design is slightly different with the first one. The purpose are different. So this is published in the Translational Animal Science. So this is the design. So we have 4 treatments. One, the lactic control, basal diet, and the poultry control, we use avelamycin. So because by that time, we cannot use the penetration anymore. So because it is already banned. So, but this avelamycin right now is the most popular antibiotic used in poultry industry. So the usually called Shumax come from the Elanco. And we use the 250 ppm. We want to increase the dosage, 250 ppm combination. And they also use 500 ppm, the combination, use a high essential oil. And this a lower essential oil supplementation. So, and also we have a 3-phase starter and a global finisher. So this time we keep the birds longer. What do you want? We wanted to see a heavy birds to see the incidence rate of the white striping and woody meat. So at Argenis to check the meat quality. So this is the design. So and then we look at the intestinal morphology, apparent digestibility and gene expression, and the ineocecal microbiota by 16 SRN sequence. And then we did the meat quality, so with the help of Argenis. So they look at the myofibril fragment index and the pH and the cooking loss and the shell force and the meat color, and also the white striping and the woody breast. Okay. So because we didn't see the difference in growth performance. So this was conducted in the University of Manitoba with the University Research Setup. It was very clean. So we didn't see the growth performance. Even we see the little bit better growth performance in antibiotic treatment. So there was no difference in the organ index, but with the meat quality, we found the shell first was reduced by the high dosage essential oil treatment. So we also lower the score for the white striping and the woody meat, but not statistical different. So you can see this essential oil reduce the shell first. That means increase the quality. And so we look at the digestibility of the nutrients. Okay, the dry matter, So essential oil low dosage improve the dry matter digestibility, and look at high dose, no significant improvement. So low in dose increase the crude protein digestibility, and the high dosage no significant improvement. So it looks like for growth performance and digestibility, the low dose is better. And for the crude fat, so both essential oil, high dosage, low dosage enhance the digestibility crude fat compared to the control group. So even we see the improvement in digestibility, but we didn't see this was not translated to the growth performance, right? And also we look at the digestibility amino acid. So generally speaking, the essential oil can increase the amino acid digestibility, as I mentioned on the previous slide. So these are not translated to the growth performance, but they still can see the digestibility of the amino acid was increased. So and then we look at the morphology. There are no difference in the vellus height, length, and the creep depth, but the ratio. So these both treatment can improve the vellus creep ratio, that of duodenum and jejunum. That means this essential oil treatment improve the gut development, had a better gut intestinal development. And also we look at some like gene special related belly function and the transport. So we see that they increase the tight junction protein expression that means have better linkage between the cells. And also we see some increase of amino acid transport expression. So which means can have a higher transport absorption efficiency, that we can see the higher digestibility. So this partially explains that. So with the nutrient transport, in your microbiota, so essential oil high dose reduce the relative abundance of the proteobacteria and increase relative abundance of the lactobacillus. This is a similar result with the first study. So with increase the number of the beneficial bacteria by the essential oil supplementation. So based on this summary, so dietary encapsulated essential oil improve the meat quality by reducing the meat share first probably, yeah. And the dietary encapsulated essential oil increase the vellus and the creep ratio in the small intestine, and they impair the digestibility of nutrient, even not translate to the growth performance. And they change the microbiota composition by increase the beneficial bacteria, for example, lactobacillus. After that, so we have the general picture about this essential oil supplementation. Looks like can bring a lot of beneficial for the birds, and then we can reduce the antimicrobial resistance and reduce the virus gene and the plasmid, right? So, and then we want to look at how this the application of essential oil to link it to human risk. So we want to check if this isolate, E. coli isolate, come from the poultry industry that can infect the human a lot, right? So if this can affect the human and this bring potential risk for the humans, so and the essential oil application can reduce the antimicrobial resistance or control this bacteria. So this can be bring the benefit for the public health. So in order to do that, we check the various potential of the antimicrobial resistant extraintestinal pathogenic E. coli from a retail poultry meat in the C. elegans model. So this E. coli is not come from the animal study. We just isolated this from the retail chicken product or feces. So to give you a brief introduction about the C. elegans. C. elegans is a worm, and so the two purposes that fit our two properties fit our purpose. One is can be infected by enteric pathogen, and a model for the innate immunity study. So it's very easy to grow and a very short life cycle, and it has very clear genetic background. It's all whole sequenced, okay? And it has greater similarity to higher vertebrates. 50% of human genes have their homologue in the worm. So that's the reason why we use this C. elegans as a model. So basically we use the C. elegans culture. So you can see just that C. elegans use the E. coli as food, that eat the E. coli and the non-pathogenic E. coli. So, and then we need to synchronize to make it grow at the same phase. And then use the test the E. coli culture. The E. coli, one is the E. coli up 50 is the control. It's a food for the C. elegans. And this E. coli KA, let's now call F4 as poultry control. And one human extra pack, EX pack, and the 29 from a chicken meat, and the 12 from a turkey meat, and 4 from a chicken fecal sample. So total about I think it's a 49 isolate. So and then we also do the whole genome sequence, and then based on the relationship to the human expect, we select the A isolate for the C. elegans study. So this for the first one, and if the C. elegans culture and the E. coli culture, and they put together, and then we monitor the lifespan or cell of the C. elegans. So based on the final genetic tree, we selected 8 exapac E. coli, 1 as a human, 3 expect from turkey meat and chicken meat. They're in same subclade with the human expect. 4 expect from chicken feces with a different number of the VGs and the ARGs. So all they have the antimicrobial resistance. These are all the antimicrobial resistant E. coli. So the first study, we see the OP50, is the liquid control, no cause reduction in the survival rate of the C. elegans. And the KAA, the E. coli KAA, and the human expect the 1 to 10A, 50 to N. So no significant reduction in survival rate of C. elegans caused by at this dosage, but they have a higher dosage, they can reduce the survival rate of the C. elegans. So it means the liquid control, this is the food cannot cause any reduction, but for the human expect and the E. coli KAA can cause the significant reduction with a high dosage. So, and then look at the study 2, and so this is isolated from a chicken. You can see the reduced survival rate of C. elegans compared to the OP50, and about 70% of worm days after 12-day incubation. And here, so this another 1, 2, 3, 4, and this 1 to 10-9 CFU per meal reduced survival rate of C. elegans compared to OP50. Nearly all the worm days after 12-day incubation. So which means this isolated can infect the C. elegans. This shows the potential to infect the human as well. And especially these are also antimicrobial resistant E. coli. So here, the other strains also show the reduction in the survival rate of C. elegans compared to OP50. So you can see the result. So based on study from this one, so the XPEG isolate from the poultry meat tested in this study may have a potential for human infection. And interestingly, the number of the VGS I didn't show the result, also may directly affect the severity of the host infection. So AMR expect from retail poultry meat or from the industry, like the farm, may present food safety risk. And the use antibiotic alternative to remove the antibiotic can reduce the antibiotic resistance development. So it has a benefit for human, for public health. So based on the 4 study, we can conclude the encapsulated essential oil can be used as an antibiotic alternative through controlling the NE, improving the intestinal digestive absorptive function, and modulate the gut microbiota and in the broiler chickens. And the encapsulated cinnamaldehyde essential oil, or citra, can also reduce the resistance level of the antimicrobial and lower the prevalence ARGs and plasmid MLSTs in the E. coli, which could reduce the risk of expect infection in humans. As this expect actually from turkey meat, chicken meat, and all feces show the zoonotic potentials. So these are very interesting. So based on that of the future study, future research direction, I think a complete assessment to investigate the toxicity and safety of the encapsulated essential oil in broiler chicken. Especially, as I mentioned before, because the 2 essential oil can already approved to use in the feed. But the CFIA put the maximum level. I can't remember exactly the concentration. They're about 20 ppm. But the study were much higher. So you want to get approval from the CFIA, so this study is quite essential. Or you apply for the drug permit from Health Canada. So that's the reason why 2 product based these 2 essential oil is get approved for the Health Canada. Health Canada has a much higher allowed level. So in CFIA is about 20 ppm. So if we want to get the CFIA approval, so this study is needed. Okay, and the on-farm trial, because in Quebec and the UM, they're also research station, right? University research station. So the on-farm trial is still needed to validate the effectiveness of the encapsulated essential oil, control the NE and impact of the chicken growth performance, and fitness into the current system commercial production broiler chicken. So actually, it is already done, but I haven't get the result yet. So based on the feedback, I think was quite good. So because the on-farm study, they use a large number. So we used the 30,000 birds each time, repeat 7 times. So because you cannot set up negative control because only use the antibiotics and also the essential oil treatment. So this actually is already done, but we haven't get the result yet. And I also want to thank the funding agency like Agriculture Food Canada, Innovation CFI, and also WSF for the in-kind support, and the NSEC, and the Genomic R&D Initiative, and also Canadian Poultry Research Council, so for their financial support for this project. Actually, this is really the teamwork from different locations, Quebec, Guelph, and Manitoba, and the different research group. For example, I want to thank my group, and for my research associate and the graduate student, especially Chengbo, this my former PhD thesis student, and this is his whole thesis. And also thanks the group from Dr. Geng from AFC Guelph, so for the development of encapsulated essential oil, and also thanks for the Dr. Lala's group for the whole genome sequence, so you can see a lot of work. So also it's quite expensive as well. And also thanks for our meat scientists, the Argenis, for the measurement the meat quality. So, and also very much appreciate for that. And yeah, that's all my presentation. I very much appreciate for your listening. There any question? No.
Thanks, Chengbo. Well, we have a few minutes for questions. So if you have questions for Chengbo, this would be the time.
In your opinion, what could be the reason for decreased resistance in older chickens? In your study too.
So you mean the essential oil supplementation or?
Yeah, because you mentioned that as the chickens got older, you got lower results for increased resistance.
Yeah.
So, in your opinion, because you mentioned that you don't know what caused it, but I just wanted to know what do you think could cause it?
Yeah, well, I think probably the one reason because usually the birds are more susceptible to the disease, right? So maybe the microbiota is not stable yet. With increased age, so the microbiota composition is stable and more stronger, right? So that maybe this is one of the reasons. But as I mentioned, we don't know yet. But this may be the one reason for that. Yeah.
Do you think that's a research gap?
Yeah, potentially. Yeah. So yeah. Mm-hmm.
Any other questions for Chengbo? Christina?
Yeah, I see that you have used the chemical reaction to make the coating between the soy protein and also the polysaccharides.
Yeah.
Did you check the acrylamide amount? If you have any acrylamide information?
So you mean so your question about the procedure macroencapsulation, right? Yeah, so I didn't show much about the procedure here. Basically, they check the morphology, particle morphology, and use the scanning and like a microscope, electronic scanning microscope to check the morphology of particle, and also they check the loading capacity, encapsulate efficiency, and also to check the in vitro release or the in vitro release and the in vivo release. For example, how much released in the stomach, how much released in the duodenum, jejunum, and the inu and the colon, right? So this is already validated in the previous study already.
So I guess the question was the Maillard's reaction where you combine the protein and the sugar, right, the polysaccharide.
Yeah.
I think Christina was asking if you considered
Acrylamide.
acrylamide, right? Was acrylamide considered?
Development. If development of the Maillard reaction is maybe the development of acrylamide.
Or that could be the good option because that reaction actually make the particles stronger, and they also the uniform of the particles are better, and right, so hold the essential oil much better than just mix the soy protein and polysaccharide. Yeah, but maybe I don't know too much about that. Maybe potential to do that. Yeah.
Well, this is going to be sounding like a really bizarre question, but we feed essential oils to honeybees for growing lights and things like that, diseases. And we winter bees in a building. One of the producers asked me yesterday about the odors that are present in the building for feeding bees essential oils. And is there any health risk to being in the building? So I'm wondering if when you encapsulate this material, you're feeding it to chickens in a broiler barn, is there any smell of the essential oils that's given off, or is it pretty much seem like it's odor-free in the barn?
So sorry, I didn't get the question.
So basically, is there any smell that comes from application of essential oils as a feed additive in the barn or in the environment? Because apparently that was noticed, but I think yours is encapsulated.
Yeah.
But you also mentioned
does the encapsulation contain that entirely?
Yeah, can it contain all the odor that would come from essential oils? Because it's a pretty strong odor, right?
Yeah, that's a good. I think it's a good question. That's one of the reasons they want to encapsulate it, right? So, the low concentration of essential oil, the smell is good. If it's too strong, also not good, right? So, and I think I also get a sample. Look, smell is okay. So once you consider inclusion rate in the feed, right? Usually about 100 gram or 200 gram per metric ton. The inclusion level is very low. So as the smell is not a concern after encapsulated. Yeah.
Good question. So you get that oregano oil, for example, you use it right away, you're like, wow, this is strong stuff.
Yeah.
Any other questions for Chengbo?
No.
Yes, Marquez.
My question is related with the antibiotic resistance. When you said the incidence of the antibiotic resistance, the phenotype part,
Yeah.
with the different antibiotic, but you determine the percentage of E. coli strain that can be multi-resistant.
So you mean for individual antibiotics and also multiple?
Yeah, so because sometimes one strain can be multi-resistant.
Yeah.
So do you determine if any strain is multi-resistant more
Yeah.
than 2 antibiotics? Or because of the time I realized that there was individual antibiotic, no matter the strain. So
Yeah, based on the phenotype, I think the strain usually had multiple antimicrobial resistance, multiple. Yeah.
All right, was there any other questions for Chengbo? Anything else? Didn't realize how big this sector was, the industry itself. So if there's no other questions, I wanted to ask you, are essential oils already used? And you mentioned some of them have been approved. Do they make up any part of the market yet, or is this just starting? Where is this at economically?
Okay. So, as an effective antibiotic alternative for you should be work first, right? And also the economically feasible, and the friendly to the environment, and accepted by the customer, and also can easy to apply. So one of the reasons I'm making encapsulation to make it easy to apply. So actually, the industry in Canada and also US, there are many products based on essential oil already, non-protected or encapsulated already. They're used in Canada and US. So you think about the Canada, the broiled chicken, and the rest without antibiotics already about 20% in the broiled chicken, they have antibiotic-free production. And the US is higher, 45% of the rest without antibiotics. And in Turkey, the hens usually no antibiotics, and the terms, in Canada maybe 50% with antibiotics, 50% without antibiotics. So for layer, we don't use, right? So I think they already have the product in the industry, but so that means the rest without the antibiotic production is feasible, but it's terms of cost. So we try to reduce the cost. Even the feed additive, we try to reduce the feed cost. It's feasible, but because who will pay for that? So we try our best to reduce the cost. So one of the reasons encapsulation is also to reduce the use dosage, right? We reduce the loss during the processing, during the application, so which also can reduce the cost. So all about the cost, I think RAW is RWA, forget it, rest without antibiotics is feasible, in terms of cost management and right.
And just one last thing, you said these essential oils have flavor, right? If you were to consume them.
Yeah.
When you guys did the meat study, did anybody taste the meat? Was there any flavor of essential? Is there a flavor study?
Flavor, I didn't, even though we didn't evaluate the flavor, but during the process cutting and doing those analysis, I didn't detect any aroma.
Yeah.
Yeah, because we have people in the faculty that do flavor type work, right? So I was just wondering if they were involved. Anyways, just a question.
Yeah, I worked with the feed flavor company before 2.5 years.
Okay.
So basically, the feed flavor also have it's about the concentration. Use very low concentration of the flavor that can improve the feed intake. So based on concentration, it's too high, it's no good.
Yeah, we all had these eggs that taste weird.
Yeah.
Anyways, if there is no other questions, we'll close the seminar. Thanks again, Chengbo.
Yeah, thank you everyone.